these are actually slightly enhanced. Instead of having the usual chemistry that we all live off of, there have been a couple of key substitutions made to how these things are put together. And they've been carefully crafted over about 2 or 3 decades of work and polishing by chemists to make subtle changes that enhance the properties of these short snippets of genetic code so that they last longer in the body, they have better penetration into tissues where we want to deliver them, and that they're better and where they're better tolerated. So it's not exactly our native RNA, but it's close. How are these ASOs used? Well, we can actually bring ASOs to bear on different genes to elicit different effects. One effect that ASOs are well known for is for changing what's called the splicing pattern of genes. These are called splice modulating ASOs. And I tell you about this only for historical context. The first really successful ASO that showed us that this kind of drug technology could work was FDA approved about 10 years ago, December 2016. And it was a splice modulating ASO that changed the assembly instructions for a gene called SMA, the gene responsible for a condition called SMA, or spinal muscular atrophy. And that was the first application of this technology that put this type of drug on the map. It was a remarkable drug that showed for the first time that a pediatric neurologic condition with very severe consequences could be rescued with early application of a drug like this. Another common application that we use ASOs for is for gene knockdown. These use a class of ASOs that are called gapmers. The details aren't important, but the point of this type of ASO is that they bind to their targets and they actually will inhibit, they will suppress or destroy their targets. They will actually bind to a particular site on the RNA, and they trick the cell into thinking that that RNA is an invading virus. And they trick the cell into then degrading that quote unquote invading virus, chewing it up, destroying it, and making it unavailable to be turned into protein. And this is the key approach that we're actually using for ATP1A3 that has been used for many other drugs too, which I'll explain in a moment. You may hear of other applications of ASOs with terminology like tango, that sounds like fun, exon skipping, et cetera. Those are specialized applications that you can ignore. These two, the splice modulating and gene knockdown approaches, are the workhorses for how most of this technology is most commonly used. Using an ASO gapmer, we can silence mutant copies of genes that are interfering with normal function. And that's the key, the holy grail in ATP1A3 associated disorders. Mutant copies of this gene that might be interfering with normal function, we would like to target for destruction so that the good copy can continue to work unimpeded. How do we do this? Well, each of our copies is present in our genomes in 2 different copies, one inherited from mom, one inherited from dad. And in kids with ATP1A3, one of those copies is defective. The copy that's normal is called the wild-type copy, and that's making normal RNA. The copy that's mutated may have a single letter change in it that results in it poisoning the downstream complex. And so the formation of a mutant protein by that mutated copy is what leads to interference that then actually has impact on the cell, even on the good copy of the gene. If we can use an ASO to selectively target the mutant copy, we can destroy that copy. And then by chewing that up into little bits, those are the fragments, multicolored fragments on the right, we can keep it from turning into protein, and we can hopefully now restore normal healthy function or improve function of the cell and let the remaining good copy of the gene do what it should be doing without interference. How are these drugs administered, if you can pull off that magic that I just described? ASOs are given to patients with severe neurologic disorders via intrathecal dosing. Intrathecal means it's given into a structure anatomically that's called the thecal sac. It's a sac that envelops our spinal cord and brain. It's more commonly known as the spinal tap. And it's maybe not the most fun procedure that you can have on a Friday afternoon, but it's not difficult either. It's something that actually can be done with a little bit of local anesthetic, a little bit of pressure, and maybe a 30-minute procedure to have an injection done. I don't want to oversimplify it, of course. A healthy adult can have this done in 30 minutes and be in and out the door. Children who are having this procedure do need anesthesia, and there's a more complicated piece to it, but that is the basics. And using this type of technology and that type of routing administration, there have been multiple FDA-approved ASO drugs in the last 10 years. On the left are the full list of all of these fully FDA-approved ASO drugs that are on the market today. Two of them are highlighted in yellow. Those are the two that have been approved for brain indications. One of them, the first one, is a drug called Spinraza or nusinersen for spinal muscular atrophy. The second is a drug for ALS called tofersen. This is Lou Gehrig's disease. On the right, in the yellow box, are a series of additional ASO drugs that are currently in clinical trials for neurologic diseases, some of which are like ATP1A3-associated neurologic disorders. And the point of this slide is to say that there's a lot of activity and a lot of now growing experience using this type of drug given in this way for kids like those in this room. Just a word about the actual clinical trials. So these clinical trials that are currently running for many different diseases involving the brain and central nervous system. As we mentioned, they're given via lumbar puncture or spinal tap. These are often done in operating rooms under sedation for kids. Dosing generally occurs every 2 to 3 months with an early phase where we might be giving more doses to first test doses to build up the dose levels, but after first few loading doses, the maintenance period is every 2 to 3 months. And these clinical trials for ASOs often include additional study visits. It's not just coming in and having a dose, but of course, you have regular visits to be reviewed, interviewed, examined by physicians to see how you're doing. And so in addition to coming in every 2 or 3 months, you may have study visits every several weeks. The costs associated with these clinical trials are typically covered by the study sponsors, which are classically pharmaceutical companies and biotechs. And some of these some of these fees are charged to insurance companies if they overlap with things that your children are already doing as a part of routine care. But this is sort of how the mechanics of all of this works. So how do we find ASO treatments for children with ATP1A3-associated diseases? Well, first, we have a screening process. And the screening process might look like this. Shown here is a segment of the genome, say, overlapping ATP1A3. And shown in the colors, those colored little spikes are different mutations in ATP1A3, some of which may be causing disease. Those mutations become little molecular handles that we use to put ASOs over so that the ASO is binding to the gene at the site of the mutation. The normal copy of the gene lacks the mutation, so the binding is imperfect. The mutant copy, it's a perfect fit. And the ability to discriminate between those subtle changes is something that we are looking for from the ASO so that it will be selective for just the mutant copy and retain the good copy. Designing ASOs like this on paper is easy, but then to actually screen them, we have to screen them in cell lines, which we laboriously work to develop with partnership from all of you. If you have blood samples or skin samples taken, we are using these to derive cell lines that we can use to create a bank of these cells that we can keep in our freezers and grow and use for repeated screens. And we apply the drugs that we've designed to those cells in little dishes, and we screen to find those that will selectively knock down or reduce levels of the mutant copy of the gene while leaving the good copy intact. What does this look like? It looks hopefully something like this. If we were to plot the levels of gene expression that we can find in the cells, in the patient cells that we're growing, and we can plot 2 different levels. We can plot one level for the normal copy in blue, And we can plot another level for the mutant copy in red. What we hope to see is that as we increase the dose of the ASO drug, from left to right, we're marching up increasing doses of the ASO drug, that we are able to see reduction in the expression levels of the red line, the mutant copy, with relative preservation of the gene copies of the normal copy. And by looking at RNA levels, that's our first indication for an ASO drug that looks promising. Well, what do we do after that? Well, that's just finding a change in a number, like the RNA copies of the gene in the cell, is just the first step. We also look for evidence of efficacy in terms of making the cell healthier. Is it actually doing what we want the drug to do to the pump, to the cell, to animal models of disease? And these are critical validation points to make sure that the reduction of the mutant copy is actually having the effect that we want. How do we do this? We may work with scientists like Kathy in the back, who've pioneered biochemical assays and readouts for measuring activity of the pump to make sure that cells now treated with this drug have overall better pump function. We might work with scientists like Madison in my lab over here, who presented yesterday at an internal meeting about using stem cell-derived neurons from patients with ATP1A3 and measuring how they communicate with one another in dishes and looking for evidence that the drug that we give helps them communicate better in more normal ways. And we might work with scientists like Marcus in the back and Kat Lutz, who have developed mouse models of this disease that copy many of the symptoms of your children and to see whether or not their symptoms get better. We also look at, obviously, safety. Showing a drug might have a beneficial effect doesn't mean it doesn't have a bad effect. And so it's important for us to look at using cell lines and animal systems and increasing levels of scrutiny to make sure that these drugs can be tolerated. The good news is that we have a lot of experience with this type of drug, this ASO category, with the maybe 10 drugs approved and another 10 trials underway. The FDA has seen many, many examples of these safety packages. And so there's a fairly standard way that we put together those packages, looking for evidence of immunotoxicity first in human cells, for first doing preliminary screening studies in mice, where we administer the drug via a needle to the brain of the mice and see whether or not it has any ill effects. And then we do multiple repeat dose studies that might last up to 3 months in rats. And there we'll use something more like human dosing. We'll use spinal taps, intrathecal doses, repeatedly in those rats to make sure that it's well tolerated. Typical screening for the ASO programs that have entered clinical trials typically involve numbers like this, where you first design and assess computationally ASOs that might be useful to you. You might look at hundreds to thousands of those. You then manufacture those that look most promising. You apply them to patient cells in little dishes, and you might screen dozens to hundreds of those candidates. From those, you hopefully find, say, a dozen that show some promising efficacy, and then you do downstream testing to find those that are both potent, selective for the mutant copy, don't have off-target effects on the normal copy. And you will subject those to the safety screening through cells, mice, and rats, as I described, and then you package up 1 or 2 of your favorites to submit to the FDA to hope to open a trial. And this is the type of process that we are trying to get through for your community in our programs. What does the FDA filing look like? Well, once you've found your clinical candidate, now it's time to put all of your testing really to proof. And so we have to do careful clinical grade manufacturing of the drug with the vendor that will make it in the clean room. We have to do a careful formal FDA registered toxicology study that's done typically by contract research organizations that have auditing and quality control metrics to make sure that the data submitted is clean. We have to apply to the FDA, and then if given the green light, we can start opening a clinical trial. So I've given you an overview, of course, and along the way, I've described all the things that we are in the midst of doing right now with many scientists around the world. Our current status is that we've successfully set this screen up and we're defining the critical parameters for success. We are asking really critical questions like how much knockdown can we elicit with ASOs targeting ATP1A3? And we've seen ASOs that are capable of knocking down 50 to 80% of the gene of the basal level of ATP1A3 in cells. We are working hard to make those selective, but we're seeing that potency is something that we can achieve. We're asking critical questions like how much knockdown of the mutant do we actually need to have a beneficial effect? And the important complementary question, how much knockdown of the good copy might we be able to tolerate? No ASO drug is perfect and will always have some off-target knockdown. So these are really critical questions to address. How are we handling this? We're working with scientists to address these questions using a variety of disease model systems, ranging anything from looking at frog eggs, xenopus, oocytes, and injecting them with different amounts of normal or mutant ATP1A3 to see how much pump activity they have, to patient skin cells, patient neurons, as I've described, and the mouse models that I mentioned as well. We're currently somewhere about here in the pyramid. We have evaluated this for a pilot indication. We picked E815K as a mutation associated with the most severe form or one of the most severe forms of ATP1A3 associated neurologic disease for which we have the most leverage with the FDA to move quickly. And we've identified ASOs and screened them in patient cells and are working to find those that have the appropriate potency and selectivity. This diagram makes it sound like there's just only one way to go on the diagram. You just start at the top and you work your way down. I will say that science never works that way. It's never linear. And there have been times when we thought we were all the way down at the second level and ready to start safety studies. And then there have been realizations where we've had to stop and say, wait, we have to backtrack a bit and then go up a level and rescreen and retest. And we are in the middle of those retesting and rescreening cycles right now. But as we're doing this, we're building the plane as we're flying it, developing all of these assays and the critical parameters for what success looks like. And we're really optimistic and hopeful that we're going to land something that might be a clinical candidate in the not too far future. So with that, just returning back to the team of folks that we have the pleasure of working with, if you see any of the folks on the slide here at the meeting, feel free to hunt us down and chat with us. We get a lot out of coming here and meeting all of you. This is really meaningful work to us, and the chance to hear your thoughts on it and what it means to you is really motivating to all of us. Thanks so much.
I just have a question around the FDA-approved ASOs. Are there any other agencies or authorities that have approved ASOs?
Oh, that's a good question. So all of the programs that I mentioned on the slide were first approved in the US. And then several of them then have also then been registered and been more broadly approved in multiple other countries. The initial drug in 10 years ago, Spinraza for spinal muscular atrophy, has been approved by multiple international regulatory authorities and is in broad, although not completely widespread use. It's still pretty expensive, so some countries are still negotiating and don't have complete access to it.
In your experience after the FDA approved it, how long would it take for other agencies or authorities? So
in my experience, it happens relatively quickly. The major markets, of course, which we'll leave out some, are obviously US, UK, Europe, the Middle East. There's usually a first wave through those authorities, and then broadening out to more global distribution takes a little bit longer.